Each year, Microsoft Research hosts hundreds of influential speakers from around the world, including leading scientists, renowned experts in technology, book authors, and leading academics, and makes videos of these lectures freely available.
Okay, so today we have Brendan Fry with us, back to give us another fascinating talk. He has visited us many times before, and most of you know him already. But just as a short word of introduction, Brendan is a seasoned machine learning researcher, probably best known in machine learning for his early adoption of message passing algorithms on loopy graphs. And he has proposed this really interesting way of describing probability distributions using factor graphs, which are really amenable to message passing. And he's been working on that for a while. Most recently, he had a paper on affinity propagation in science, which is really a clustering algorithm, but it's using similar intuition, message passing intuition. But recently, Brendan has been probably better known for his work in biology. His work has been published basically in every major journal as of last week or so. The final frontier has fallen after Science and Cell. Now he has published a paper in Nature, and he ended up on the cover of Nature. So he will talk about his research from that journal and all the research that preceded it. Now, I should also say that he is working with some excellent people in Toronto, the biologists. So that group is really awesome. The work that they're doing and the postdocs that they have been producing for the rest of us are really excellent. So.
Thanks. The talk today is sort of, as you can tell, partly biology, partly kind of software design or information theory, if you like. So how many people recognize this picture in the background? Does anybody recognize that? Yeah, what is it? The Enigma machine. Yeah. So this is a device used to encode messages, and it was used a lot during World War II, but it actually has a history long before that. It wasn't used to encode messages of this sort, species, human, tissue, brain, chromosome 12, but that's the connection to today's talk. In fact, when we were invited to submit artwork for the cover of Nature, my postdoctoral fellow, Josef Barash, managed to get this image. And he was quite resourceful. He contacted this old fellow who went to a military museum late at night and was able to sort of sneak in there and slip this piece of paper in behind the glass barrier and then take this picture. So very resourceful postdoc. So I thought I'd make a connection to something even more recent than our Nature paper, which was published 2 weeks ago. 5 days ago in Science magazine, Craig Venter's group published a paper describing for the first time synthetic life, which they used a computer to design a 1 mega base pair genome, and then they inserted it essentially into a cell, a host cell. There was no other DNA in that host cell, and then the resulting cell was able to, formed life. So it was able to divide successfully, so reproduce. So first synthetically generated life. So what does that have to do with my talk today? Well, I think this is a nice demonstration of where we're at in terms of being able to use computers to design sort of codes, in a sense. The genome is kind of a code. But what do we need to do in order to understand how humans work, or more complex organisms? And that's what I'll be talking about today. So here's a nice illustration of what I'm getting at. The number of genes is not all that matters. The balsam poplar tree has 45,000 genes. And the human, and this is a picture of my baby, has 22,000 genes, and yet it has several tissues that are much more complex than the balsam poplar tree, notably the brain. And I'll actually come back to, and I'll justify that statement later on in the talk and show you some very interesting examples of what's going on regarding neural network wiring. I want to give you a bit of a sense of history in terms of the community and what's been going on, because not everybody here is knowledgeable about the field. So DNA was discovered in the 50s, and this human genome was, the sort of near final draft of the human genome was assembled in 2004. And so we talk about that being a book containing about 3 billion letters, AGCT. and containing around 20,000 or 22,000 genes. Okay. So this is an interesting quote. Genome bought the book, hard to read. So this is what Eric Lander's comment on the genome was. Yeah, so you can take a look at this 3 billion letter book, but it's hard to make sense of it. What's going on? So that's what we're going to be talking about today. So let's take a step back. How do genes actually encode genetic messages? Genetic messages are the things that flow out of cells, the cell nucleus, and then encode proteins, which carry out functions in the cell. So messenger RNA. And here's a very important picture in this story. And how many people recognize this picture? Okay. So this is an electron micrograph that was taken in the 70s in Phil Sharp's lab. And what it's showing is essentially the notion of the exon and intron. So what they did is they took a piece of DNA, and they took the corresponding messenger RNA, and then they did some reverse transcription, and then hybridized the two together to see how they matched. Okay, so how does a messenger RNA or the genetic message match with the DNA? And what they found is that there was, of course, a good match for long segments, but then there were parts of the DNA that did not correspond to segments of messenger RNA. And so these loops do not have a matching template or matching sequence in the messenger RNA. And so it was realized that not all segments within a gene in the DNA actually end up in the protein. Some of them are left out. And those are called introns, and the segments that are retained are called exons. So it's interesting that they actually didn't receive the Nobel Prize for this discovery until the 90s, 1993. Seems like it's a pretty fundamental and important discovery, right? But I guess the consequences weren't really understood until a bit later. and today, that's what we'll be talking about in greater detail. So how does this work? We've got DNA with exons and introns. Somehow these introns get removed. What's the process by which this occurs? I'll show you two different pictures of what's going on. The first one, if we look inside, say, the human brain, look at a single cell, a neuron, look in the nucleus, look at a particular chromosome, and look at a gene that's being expressed within that chromosome. This is the sort of biophysical picture of what's going on. So there's the DNA double helix. The two strands are pulled apart within this big protein complex. You can't really see what's going on, but they're pulled apart. And then one of the strands is actually copied or transcribed into RNA. So this little tube here that winds around is an RNA molecule. Now, if you look closely at what's happening, concurrently with transcription, the exons, which are shown in blue here, and the introns, which are shown in green in this picture, are analyzed by this complex called the spliceosome. And the spliceosome recognizes the introns and then cuts them out. Okay, so concurrently with transcription, the RNA molecule is being fed through the spliceosome complex, and the introns are being cut out, and the exons are being glued together. So you end up with an RNA molecule that has no introns in it.
The transcription is actually a complete replica of the DNA strand, in which that's then processed, rather than sort of the DNA folding such that the ribosome sort of skips over segments.
Ribosome. We're talking about, sorry.
Oh, sorry, that's protein.
Yes, that's right.
Okay, sorry.
Yeah, that's right. So the ribosome would be analyzing the RNA molecule. Yes. Mm-hmm.
But so it's not the case that the protein sort of folds up and then the thing that's doing the transcription sort of skips off the loop of DNA. It's that the DNA is really fully unwrapped. It's all transcribed to mRNA, and then the mRNA is cut.
I see what you're saying. Yes.
Okay.
So this process here, right? So you're sort of saying that this process implies that there's kind of these static machines that are sort of assembled and working their way along the DNA, when in fact they're actually being disassembled and reassembled. Yeah. All right, so let's look at another picture. This is just a more abstract a version of what's going on, kind of one that would appeal more to sort of sequence analysis type approaches, or people who do research on sequence analysis or information processing. So you can think of the DNA as just a discrete symbol sequence. We think of a gene as consisting of certain human-defined elements like the promoter, which contains some code words that are necessary for proper transcription, and then these exons and introns. And then in this nice box and line picture, we think of transcription as basically copying the segment of DNA, and then splicing as the process that takes this RNA and removes the introns, all this stuff here, and we're just left with the exons, which then are translated into protein. Okay, so that's the box and line picture of what's going on. This kind of hides the fact that really these two processes are going on concurrently. Yeah?
So, that's something to talk about. If there's something going to be removed, why is it there in the first place, the introns?
Yeah, so the question is, if it's going to be removed, why is it there in the first place? That's a very good question, in fact. And it turns out that the introns contain a lot of important information. And that's the code that I'm going to talk about today. So the introns contain, of course, they don't contain sequences that are going to end up in the protein, but the introns contain code words or instructions, if you like, that tell the splicing machinery how to process the molecule so that it can achieve different kinds of levels of complexity. So, for example, you can have a messenger RNA that does one thing in one tissue and a different thing in a different tissue, all because of the code words or the symbols that are embedded in this intron sequence. Yeah.
Where does that information get embedded into mRNA?
Pardon me?
Where does that information from intron get embedded into mRNA in order to do all other things?
Oh, well, that would be evolution.
Oh.
Yeah.
So then, are the introns, is that the like junk DNA that people refer to?
Yes, they call it junk DNA, even though it's not really junk DNA. Yeah.
Right, but that is the intron.
Yes, that's right. Yep.
Sorry, the previous question, the way I interpret that is, how does the information from the intron make its way to the step in the mRNA that's different?
Oh, I see. Okay, I thought you were saying how is that information generated? So it's generated by evolution. So mutations occur in intronic elements. Actually, there are other kinds of things that could happen. But one way you could get information embedded in the intron is just through evolution. But how does that information control what's going on in terms of the splicing? We'll talk about later. Yeah, it's a good question, and it's exactly in line with.
mRNA and protein is one-to-one corresponding.
No, turns out not.
Oh.
So let me tell you about that. So the standard picture, the central dogma, so-called central dogma of molecular biology, it looks like this. So you have your two exons, and you have an intron. And so now you're wondering, how does this process occur? So there are code words or symbol sequences or motifs embedded in the intron and exon sequences, and these recruit proteins. So what will happen is a protein will bind here, another protein will bind here, another complex comes along, and these two proteins get pulled together, essentially. The intron is looped out now and folded back against itself, and there's a junction formed here, and then the intron is cut away, and the two exons are joined together. So this process of splicing, getting rid of the introns, involves proteins that are present in the tissue, in the nucleus of the cell, binding to the intron and exon sequence, and then essentially attracting each other. Or there's a conformal change in terms of the protein structure, and that enables this cleaving of the sequence and essentially gluing together, if you like, of the two exons. Yeah.
What is the proportion of exons to introns? Is it known?
In terms of overall sequence, it's I don't remember exactly, but typical exon lengths would be about 100 nucleotides or 100 base pairs. That's sort of a median length of the exon. Introns could be, they range from, well, I guess a median length would be something like 10,000, something like that. You can have introns that are very long, 1 million bases, or short ones as well.
So where does intron go after it gets cut?
Actually, I don't know. It eventually gets degraded. So concurrently with all of this stuff, there are proteins that go around gobbling up stuff, just breaking them down into the constituent letters, essentially. So you get all these A, G, C's, and T's floating around, and then they get used for other purposes. But it may be the case that once it's removed, once it's spliced out, perhaps it has some kind of an effect in terms of regulating what's going on in terms of transcription or splicing or other processes. Like, for example, there might be a sequence in here that binds to some other sequence in another gene, preventing that other gene from being properly transcribed. Something like that could happen. Yeah. And one thing I've learned about biology is anything goes. So there's sort of central dogma. That's what they call it for good reason. And basically anything goes. So whatever idea you come up with, it's probably worth studying it. Yeah.
Since it's folded, it will degrade slower.
Yeah.
So it probably does have a function.
Yeah. true. Yeah.
So the 3D structure of DNA is determined by mRNA or by intron? Exactly. What particular form of the 3D structure?
What we kind of think of is a DNA being separate from the RNA. So the DNA structure is independent of RNA.
Oh,
yeah.
That's not something you talk about. No.
Yeah, that's right. Yeah, the whole idea of the chromosome and the structure of the chromosome and the DNA within it is a whole other research area. Now, coming back to the question, so does one gene encode one protein? The answer is no. So it's an assumption that's commonly made, but it's quite wrong. So here's why it's wrong. There's a process called alternative splicing where you have, let's call it an alternative exon, and the reason for calling it that will be clear in a moment. So there's an alternative exon, 2 introns, and what can happen is there might be a sequence feature, a motif, that binds to a neural-specific protein. All right, so in neural tissue, a protein will bind here, and perhaps that protein will interfere with splicing. So in neural tissue, what will happen is we'll get a splicing protein binding here. Instead of the other one binding here, it'll bind over there. Okay, and now what will happen is we'll loop out both introns and the exon. And in the neural tissue, the exon gets skipped. Now, in a non-neural tissue, that protein won't be expressed, and so it won't bind to that feature, and it won't interfere with splicing. And we'll get a version of this gene called an isoform, different form of it, that includes the exon. All right. So this process has been known to occur for about 15 years, but the question that's sort of perplexed researchers for the past decade is how important is it? Is it ubiquitous? Is it something that just sort of happens once in a while? Is it just a noise, a noisy process that has no function, or is it important? Before talking about that, I just want to give you a glimmer, a sense of what's going to happen later in the talk in terms of when we try to analyze data and look at the numbers. What we're going to think of is percent inclusion values. So what I mean by percent inclusion or percent exon inclusion is if we look at these two possible isoforms, one that includes the exon and one that doesn't include the exon, and we look at two tissues, say a neural tissue and a non-neural tissue, we can think of a percentage for that isoform. So 90% of the isoforms include the exon, 10% don't include the exon, whereas in this case in neural tissues, 20% of the isoforms include the exon and 80% of the isoforms don't include it. Yeah.
Neural and non-neural? Is it also liver and non-liver?
Yes, yeah, or it could be neural, liver. It could actually be one of 200 possible cell types. Yeah.
So for neural, are you talking about the neuron in the brain or
yes.
Outside the brain?
Yeah. Oh, sorry. Repeat the question.
The neural tissues.
Yeah.
Do you refer to the cells in the brain or in the peripheral neural system?
Good question. Yeah, so when we say something, I'll clarify what I mean later. But when I say neural tissues, I might mean sort of a grab bag of neural tissue. So that would be brain tissue, but maybe some spinal tissue. Things mixed together. Yeah. Ultimately, really, what we'd like to be able to do is measure these things for individual cells, right? We'd like to be able to sort of track what individual cells are doing, but we don't have that technology yet. So I want to make two main points in my talk. The first one is this. The genome plus alternative splicing leads to a radically different view of what's going on. Okay, so I've described to you what alternative splicing is. The question is, does it really amount to anything? And then the second thing I want to talk about is our contribution. So how wrong is this assumption? So I'll actually give you three different answers. One of them is by the numbers. So this is some analysis that I did with Ben Blenko a couple of years ago using a particular technology called RNA-seq, which is high-throughput RNA sequencing. And we found that over 95% of multi-exon human genes are alternatively spliced. Okay, so nearly all genes are alternatively spliced. On average, 1 in 2 exons is alternatively spliced. Most genes undergo tissue-dependent alternative splicing, so it's not just a process whereby, for some reason, a particular gene leads to 50% included isoforms and 50% excluded isoforms, and that's true across all tissues. No, in most cases, these different isoform levels vary across tissues. So it seems to have this process seems to have something to do with regulating the generation of different kinds of tissues, so tissue development. This is quite interesting. If you look at highly expressed genes, in brain, you'll see a lot more alternative splicing than in other tissues. So this suggests that this hypothesis that alternative splicing is important for generating complex tissues might be correct.
What exactly do you mean by more alternative splicing? That you have multiple isoforms present at one time from the same gene?
What I mean specifically is if you look at highly expressed genes. So if you look at genes that have high expression levels in brain tissue, you'll see a lot of alternative splicing, so a lot of variability in alternative splicing. If you look at a gene that's highly expressed in, say, liver, you'll tend to see 100% the included isoform. So it's more...
So are you talking about just having multiple isoforms
Yes.
at the same time?
Yes.
Or, okay.
That's right. So you collect together a set of tissues, a whole bunch of different cells, of course, and then you'll see these fractions be somewhere in the middle, indicating that there's a lot of variability. Yeah.
How does the effect of having multiple isoforms impact down the transcription line? I mean, could it be the case that some of the isoforms actually don't make much of a difference downstream? Or?
Yes. So they may not be translated into protein. So that's a good point here. We know that 95% of multi-exon genes are alternatively spliced. We don't know what fraction of those actually have a functional importance. Yeah. So that's an important thing. But it's hard to, we don't really have a high throughput protein analysis techniques at the moment. So maybe one day we'll have that kind of technology and then we can answer that question.
Do you see alternative splicing very time dependent? I mean, does it vary with pH and just other dynamic characteristics in the cell, or is it more like cell type?
You know, I don't think it's very well understood. People look at people who do biochemical analysis. They'll talk about Gibbs distributions and things like that. I'm not actually sure whether that's valid, right? If you think about what's going on, you know, it's not just it's not like a box of gas or something like that, right? You got these processes tethered to the DNA and the RNA and chromatin and all sorts of other things interacting. So I'm not sure whether those kinds of assumptions are valid. Yeah, so I don't really know the answer.
So are these conclusion from experimental measurement or
Yes.
computational?
No, experimental. Well, computational analysis of experimental data, yeah. So here's the numbers. So the old traditional view would be 22,000 genes, 22,000 mRNAs, and the new view looks more like this. So the next question is I'll just fill in this timeline a little bit. So the discovery of splicing in the 70s. Recent data shows that hundreds of thousands of genetic messages are generated by this process. So 22,000 genes, hundreds of thousands of genetic messages, or mRNAs. So now, is it important? So there's a lot of it. Is it important? So we're looking at this question now. So I'll show you a few examples to convince you that this is important. So there's a set of 3 genes. They're called neurexin genes in the human, in your brain right now. These 3 genes are constantly being transcribed in different neural cells. And these 3 genes encode 3,000 different neurexin mRNAs. And these mRNAs control, essentially, how your neurons get wired together. So they're critical for different processes of learning. We don't exactly understand how this works, but we know that they're critical. And I'll show you an interesting experiment that's hard to do for human. So I'll show you an interesting experiment that was done for Drosophila, the fruit fly, which gives you an idea of what might be going on. Suppose you wanted to program a neural network. So you were going to write a C program. You'd probably use 4 next statements, right? You'd loop over the neurons in the input layer, and then you'd loop over the neurons in the second layer, and then you connect them together. That's how you'd write the software. So the brain can't do that. So how could the brain do it? How could your brain develop in such a way that it can somehow connect together neurons properly? Well, it would probably do something kind of random, right? Kind of a random coding technique, something like that, something random. Seems to be that that's what actually does go on. So if you look at Drosophila, the fruit fly, there's a gene called DSCAM, which has 38,000 isoforms. And if you look at a normal Drosophila sensory neuron, the dendritic tree of that neuron, it looks like that. So that's kind of a normal-looking dendritic tree, right? It looks like a tree. If you knock out DSCAM, what you get is self-adhesion. So the dendrites are sticking to themselves. The neuron is making connections with itself, which is a bad thing for a neuron to do. It should try to make connections with other neurons. If you introduce one isoform, so you engineer the genes that there's only one of these one messenger RNA can be generated. Now self-avoidance is mostly recovered. There's still a couple self-connections, but this looks a lot more like a healthy dendritic tree than this one. Okay, so it seems like DSCAN is somehow needed for a neuron to figure out where it should connect. And so what it looks like is essentially each neuron gets a tag, a different version of this messenger RNA, and then whatever version it gets is used as an identifier. So now it can recognize itself and avoid sticking to itself. So here's the follow-up experiment, which suggests that this is actually the mechanism that's going on for connecting these neurons together. If you look at the wild type, or the normal sensory cell, and you look at two overlapping dendritic trees, they look healthy, right? So this magenta cell is connecting to the green cell nicely. If there's only one isoform, however, they avoid one another. Okay? So that's consistent with the idea that the isoform is used as an identifier. So this magenta cell recognizes itself and doesn't stick to itself, which is good, but it also thinks that the green one is identical to itself, and so it doesn't stick to the green one. So of course the next thing you'd want to know is what happens if you introduce two isoforms and so on, and that experiment hasn't been done yet. Now, so hopefully I've convinced you that splicing is important, and it's not just sort of an artifact or noise, it's actually critical, and it's ubiquitous. So now what I'll do is talk about our research. Several other groups have been trying to solve this problem in addition to ours, so I'll actually refer to some of their work. So this is the central challenge in the field, as articulated by Chris Burge, who's the leader in the area. So an important long-term goal is to determine a splicing code, which you think of as a set of rules that can predict the splicing pattern of any primary transcript sequence. So I like to think of this in terms of like a software or a web tool, where you could literally drag and drop the unspliced RNA sequence, and then the software would tell you how it's going to be spliced in different tissues. So it'll tell you in brain you're going to get this messenger RNA, and then in liver you're going to get this other messenger RNA. That would be the ultimate goal of the community.
But the answer will be statistically correct, not...
That's right, yes. Okay, so here's a fun puzzle for you. Oops. Yeah.
Recapping, when you say in brain, that's shorthand for in cells that have a particular complex of...
That's right.
Around.
Yes.
So okay.
Yeah, ultimately what you'd like to be able to do is measure the cellular state, right? Everything about the cell. And then given all of that information, predict how it's going to be spliced. That's really what you'd like to be able to do. Of course, that's not possible currently, yeah.
So, and this is only affected by the proteins that are in the cell, or do other things, if you could specify exactly how much of every single protein is in the cell, then in theory you could solve this problem?
I guess you'd need time as well.
And how many proteins are there, roughly?
Oh, well, it's we don't know.
More than 20,000.
That's right, we know that, yeah.
All right.
Yeah, and some of these modifications, so splicing might do things like change the binding tract of a protein. So it's still sort of the protein is similar in all ways except that the binding domain has changed. So it'll bind slightly differently or in a quite different way. Okay, so this is kind of what the challenge looks like from your point of view. There's an exon over here. I've given you 300 nucleotides of sequence in the upstream intron. You have to look at that and try to predict what's going to happen to this exon. In brain, say, will it exhibit increased inclusion? Will you get more of the included isoform or more of the excluded isoform? So that's really what the task looks like. Now, importantly, in the last 20 years, since the early 1990s, people have been studying this process in terms of how it's controlled and what sorts of features or code words in the introns and exons lead to different tissue-dependent effects. But they've really studied it sort of on a one-off basis. So individual genes and individual code words like the binding site of FOX or something like that, which is a regulatory protein. And now, really, as I said, my group and a couple other groups have been putting all of this information together and coming up with sort of high-throughput, large-scale computational techniques that can assemble the information and come up with an overall picture of what's going on. And so that's what I'll talk about today. So the postdoctoral fellow who did most of the computational work is Josef Barash. He's now looking for a permanent position. John Clerico was critical in doing the experimental work. So we developed this code using computational techniques and made a whole bunch of predictions of different kinds. John Clerico then went into the lab and verified those predictions, or some of them. Weijun Gao, who worked on the software, which I'll briefly talk about later. Sandy Pan, who did the sequence analysis. So as you can imagine, we've got huge numbers of these RNA sequences. We need to extract features from them or extract subsequences that might be useful for developing this code. So she extracted that information. Xinchen Wang, who studied neurological diseases and compared them to predictions made by our code. Ofer Shai, who assembled the data set that we used to assemble the code. Ben Blenko, who's a colleague at the University of Toronto, who oversaw the experimental work that John carried out. And what else? Funding. So this is funded primarily by Genome Canada, but also by a gift from Microsoft Research, and also the Canadian Institutes for Health Research. So here's the idea. It's pretty simple. It's actually what you're probably thinking of. Input, output, block diagram sort of framework. We have our primary unspliced RNA sequence. So I'm showing 3 exons and some intron sequence. We're going to extract features from the exons and introns. Given that feature set, we're now going to come up with a splicing code. We're going to use machine learning techniques to assemble a splicing code that takes the features and the tissue type and predicts whether there will be increased inclusion of the exon or increased exclusion of the exon. So you can imagine that there will be a step where we have to come up with a feature set, and then another step where we need to learn the splicing code, which will involve feature selection, and then also figuring out how those features relate the tissue type to the predicted change in exon inclusion.
It's interesting that it's not binary. That it's like a percentage of time that this code actually causes it to be a different isoform.
Mm-hmm.
How did you account for that?
I'm not really sure. It could be that in an individual cell it's highly deterministic, and then we're looking at collections of cells. Or it could be in an individual cell it's probabilistic. Yeah. So it's, yeah.
So the intron here is defined as something with no exons in it, alternative or otherwise.
That's right.
Okay.
Yeah.
But they still can range to the millions of
yes.
pairs.
Yes.
So you're looking at the ends of them because you think that's where the splicing is happening or being blocked.
Well, that's what some biologists have claimed. The reason we're doing it is simply to avoid the overfitting problem. Like needle in a haystack, yeah. If we looked at too much sequence, of course, it would be very hard to find anything that's statistically significant. Yeah.
So at this point, just no idea what's going on in the other millions.
Yes, that's right. Yeah. Could be important or may not be.
So even if you figure out this algorithm, then it should be implementable by the brain by itself, right? So it's obvious.
Then we should.
Whatever mechanism by which you can predict what will happen.
Right.
It's actually taking place in the brain. It should be implementable inside the brain using the process itself. It's also.
Oh, I see.
It's not a constraint.
Yeah. So that's a good point. So you're sort of talking about interpretability, right? Yeah. We'll come back to that later. That's a very good point. Yeah, whenever you think about trying to understand a physical system using a machine learning or signal processing technique, where you've trained it or reduced a squared error measure or something like that, or reduced a KL divergence or something like that, the question is whether the resulting model actually corresponds to what's happening physically. And we didn't know what would happen, so we just decided to go for it and see what would happen. It turns out we do have something that's interpretable and does have a physical meaning. So we were able to carry out experiments to validate the mechanistic predictions. Yeah.
One more question about introns. How do you know that you found all exons? How do you know that there are no more?
Yeah, we don't.
Inside
That's a very good point. So really, we're assuming this is the case. We're assuming that we have this nice picture. These are the introns. These are the exons. Their boundaries are well demarked, and these exons are always present. This one's alternatively spliced. We did our best to search for triplets that satisfied this property, but of course we don't know for sure that we do have valid triplets.
By mRNA
yeah, we used EST sequences. Yeah, so mined a huge.
But you're still not sure if you've looked at all possible cells and tissues, and there could be some other things.
Yeah, that's right.
So have you thought about just using the algorithm as it is to identify new exons inside?
Yeah, we are able to do that. We can we can do it with reasonable accuracy. I think the problem is that is that what really happens is there's things like alternative splice sites. So sometimes this exon might be just trimmed off by 20 bases, and you'll get an exon that's 20 bases shorter. Okay. And all sorts of other kinds of effects like this. So really, I'm talking about a fairly restricted type of alternative splicing that's thought to dominate all the kinds of variants you can get. So about 80% of alternative splicing events are thought to be of this form. But we don't really know. That 80% might be completely wrong. There could be a lot of other variants.
You were saying that the intron has these 300 nucleotides on one end, and on the other end, it tells you that these two things should bind and should be spliced out. But inside the intron, you could have another nested pair of 300 and 300 nucleotides, which you can look for and identify a new intron inside.
Oh, I see. Right.
I mean, new exon inside.
Yes.
And then
I see what you're saying.
somehow.
Yeah. Yeah. Yeah, people have done that. So Chris Burge has done a lot of work on coming up with computational methods that are simply supposed to predict exon boundaries. So he'll use a training set of known annotated exons, train up the method, and then run it on the entire genome and predict boundaries. So in a paper that we published in 2005 in Nature Genetics, we actually took 1 million predictions from his technique and other techniques as well, 1 million putative exons. So the number that was known at that time was about 220,000. So we had 1 million, so about 800,000 possible new exons. And then we used microarray data to try to figure out whether there were any new, well, actually we're looking at two different things. Any new genes, so not accounted for by the 22,000. And then the other question is any new bona fide exons. And we found about 40,000 new exons. So if we were able to predict out of these 800,000 exons, about 40,000 of those actually being bona fide new exons. But we didn't find any new genes in a sense. What we showed is really that there's what you see is different kinds of structure, different kinds of transcript structure, but not really new genes. Okay, I better continue here. So here's the idea. We're going to measure these three things, come up with a data set, and then infer the splicing code. So what about the feature set? So I said we're going to come up with a big feature set, and I'm not going to go into detail just based on the vote I took at the beginning of the seminar. You're not familiar with a lot of the proteins that are involved, so I'll just give you a high-level overview. So we have 1,014 features. 171 of these correspond to features that previously have been described. So people have characterized these and published them in papers in the past. We proposed a set of 326 new motifs that were computationally identified. 460 short motifs is what we're calling them. These are really region-specific counts of very short motifs, 1 to 3 nucleotides long. So what that might mean, for example, is if you see a lot of Cs in the upstream intron here, then maybe this exon tends to be included more frequently in brain tissue. So it's just the density or the frequency of the motif that counts. And then another class that people haven't looked at before, which we call transcript structure features. These would be things like secondary structure of the RNA. So the RNA might fold on itself, and that could influence splicing. Other things like the exon length. So if the exon is very short, maybe the machinery just won't see it, right? And it's more likely to be skipped just because it's short. Things like that. Now, what about our data set? So we've got our proposed features, 1,014 proposed features. To quantify splicing levels, what we did is we measured one number for this triplet, which is the % inclusion of that exon. So what fraction of these two isoforms include the exon, the alternative exon in the middle, A. And so we have the one number now for each tissue. For each tissue that we measure, we have this one number, this % inclusion. And this is critical. What we did is we converted it to probabilities. Not just because we like probabilities, but because we needed to account for uncertainty. And that turned out to be critical for making progress in this research. So first of all, what we have is 3 probabilities. Probability of increased inclusion, probability of increased exclusion, and probability of no change, either way. And what these probabilities correspond to is a change in a given tissue relative to the other tissues. So you might notice in a particular, say, neural tissue, that its inclusion level is, say, roughly 90%, whereas in other tissues like liver and heart and eye and things like that, you have lower inclusion levels like 20%. So then you'd have a high probability of increased inclusion in that neuronal tissue. The other thing it accounts for is uncertainty. So just by looking at the data set, we might say, well, this exon looks very noisy. It's kind of hard to predict what's going on. It's all over the place in terms of percent inclusion. And so we're not really confident about whether there's an increase or a decrease in exon inclusion. And so therefore, you'd have a more uniform distribution over these three numbers. Yeah.
So you don't care how much it increases.
That's right. Yeah.
But it's some minimum amount of increase. Otherwise, you call it no change.
That's right. Sort of relative to the noise. Yeah. Noise. So here's an example. If we have tissues here and percent inclusion, one example might look like this. Okay, so you'd probably say there's a high confidence that there's an increase in this subset of tissues, whereas another exon might look like this. Okay, so kind of all over the place. And so you might say, well, look, in this tissue it does have higher inclusion, but you might also say that there are other relevant tissues, like say this is cortex and this is cerebellum, and this is hindbrain, so there doesn't seem to be much consistency in terms of the different neural tissues. So that's what that probability is taking into account. Yeah. Yeah.
Do you have much a case of the multiple exons being spliced or not spliced, where it's not a single one, but you have a one?
Yeah. So as I said in response to Neboish's question, we used EST data to try to find examples that followed this pattern. So tried to exclude. Yeah, that's right. All right, so here's the data set now. We have in total about 3,600 exons and 4 different tissue types. We actually had 27, but we essentially grouped them together to reduce noise levels. So 4 tissue types, 3,600 exons, that gives us a total of 14,000 data points. And just a little bit of notation, we'll say that data point I has tissue type CI, so that has 4 values, and feature vector RI, which is 1,014 dimensional, corresponding to the 1,014 features, and then target QI, inclusion, exclusion, and no change. And that's the data set. So now you could use your favorite machine learning technique to essentially relate the tissue type C, the feature vector R, to the target, which is the probability of increased inclusion or exclusion or no change. So what we did is used essentially, you could think of this as a multinomial regression or a logistic regression technique that takes as input not the raw features, but decision boundaries that are inferred for the raw features. So it's like a 2-layer neural net, if you like. So what we're trying to do is predict for tissue type C and feature vector R the 3 possible outcomes: increased inclusion, increased exclusion, or no change. So this is our prediction, P for prediction. And the way we model it is we use essentially e to the power of a linear combination. So these are the logistic or multinomial regression parameters, the lambdas. And those lambdas multiply with an indicator function which determines whether the feature is in a learnt region. So uppercase R is just a set of learnt regions in feature space. If the input feature is in a learnt region, then this unit here fires, or this indicator function is 1. And then we'll add in lambda. Okay, so that's the way it works. Simple, very simple. And of course, you're welcome to take a look at the data and try out your own machine learning technique if you have one that's really well suited to feature selection. And you should try to come up with something that's interpretable. That'll be useful to the biology community. But that's basically all there is to it. That's all we did. We didn't. What we did is, in fact, so that's a good question. What do we... Oh, that's interesting. Okay, quickly look at it. I'm not able to. Okay, so how do we measure? How do we measure how good the code is? So Lee mentioned a maximum entropy type approach, and what we did is we used an information theoretic approach. If you remember that example that I showed you, where the goal is to predict what's going to happen in this tissue, it's doing it again for this one. It just isn't like that slide. Okay. Okay, so what we're going to do, so we have a raw sequence, and then we have a prediction, P, for whether we're going to see increased inclusion, exclusion, or no change for the central exon in each of the different tissues. So essentially what we're going to do is use an information theoretic measure. If you get it right every time, you'll win 1 bit of information, or actually log of 3 bits of information. And of course, it's probabilistic, so what we'll actually do is measure a KL divergence between the two distributions. So the P distribution and the Q distribution. So here's the what we'll call the code quality. So the code quality is the sum over the data set, the sum over these 3 possible states, inclusion, exclusion, and no change, Q log P, so that's the relative entropy or the KL divergence. Okay, so Q log P. And then we actually normalize by what we can think of as a naive predictor. So if you just look over the entire data set and you notice that 95%, let's say that the data is extreme, so it's either inclusion, exclusion, or no change instead of the 3 probabilities. If you look over your entire training set, you might notice that 5% of the cases have increased inclusion, 5% have increased exclusion, and 90% have no change. And so that would be your naive guesser. Okay, so that's what this Q superscript S is. It's just those three numbers. And so what this code quality metric gives you is a measure of how well you're doing beyond random guessing. So if the code quality is 0, you're doing as well at predicting splicing as a random guesser would, using just the basic statistics, and otherwise you get some positive code quality.
So you don't have the training set that tells you whether there is inclusion or exclusion?
Yes, we do. That's the data.
Then you can just count how many errors you have.
And now it's there. Yes, we can do that.
Oh, so
well, because the data set is not discrete valued. It's these 3 probabilities for this reason here. So if we see a pattern like this, then we have high confidence that in this subset of tissues there's increased inclusion. But if we have a pattern like this, then we don't have high confidence. Okay, so our raw data, from our raw data, we're not able to get discrete valued measurements or highly confident measurements. We still have probability distributions.
So those are different samples of the same tissue that you observe the.
Yeah, that's right.
And then in that case, you can separate the tissues. They
Yeah,
vary a lot. You just
that's right.
subdivide.
So you have multiple neuron tissues and you notice there's a large variability, then you don't have high confidence that there's a consistent change in a particular direction. Okay, so all we had to do is learn this thing, and it's a KL divergence basically, or log likelihood. So the algorithm works pretty simply. It actually just greedily adds features one at a time. So that's what's shown on the x-axis here. Code quality increases monotonically. So that's nice. The algorithm works. I won't even talk about that because it's not that difficult to engineer. Here's what you get in terms of this code quality. So the blue bar shows the final inferred code that we came up with. And then the other bars show what you get if you use other methods. So if you use all the previously described motifs in the literature, so the known motifs, you get a code quality that's significantly lower than the one that we achieved. If you introduce just the tissue information, so you compute these 3 probabilities for each tissue separately, you get a code quality that's significantly different from 0. Not quite as good as just all the known motifs, but surprisingly close. All right. And also, if you take our inferred code and remove components, you see a drop in code quality. So if you remove those transcript structure features I mentioned, you see a significant drop. And if you also remove some other information about conservation of motifs, you also see a large drop.
So what you're saying is that even in the red curve, we'll be able to make sense out of it using other features. What the other body said that depending on the tissue, you're not able to make much sense, and that's what the first one is.
Yes, so it's like using just the identity of the tissue gives you some predictive capability. That's what this tells you.
It's not good enough for you, so but you get much more.
I don't see the connection to the red curve.
Did you say that depending on the tissue, the probabilities are very different?
Yeah, this is not given the tissue that there's a different noise variance. This is the predictive probability. So it just means that in CNS tissue, for example, there's a lot more central nervous system tissue. There's a lot more included isoforms. So maybe 15% of them are included instead of only 5%, something like that. So just adjusting those numbers a little bit gives you a bit of a benefit.
So for this kind of research, do you have the same kind of paradigm as the standard one with the training set, depth set, test set, and then you choose the features?
Most of the effort went into setting up the problem that way. Exactly. That's what the goal was. So it's a MATLAB matrix. All right, now this is kind of what we're after, right? Newtonian physics looks great. Why? because you can take a ball and you can drop it, and you can predict the trajectory. And in fact, if you drop the ball, it follows that trajectory. I even had a video to show you, but I don't have it linked in. The ball actually does follow that trajectory. So that's what we'd like to do. Have interpretation as well as predictability, okay, being able to predict things. So I won't go into details in terms of the interpretation. It's beyond the scope of today's talk. But essentially what we have is what we call a code map or a code table that shows our different features. So there's 1,014 of these. And for each feature, we show its effect in different regions. So for example, if you look at CUGBP, which is the binding site of a particular protein, and you look in the upstream intron, you can now look at different tissues like central nervous system tissue, muscle, embryonic tissue, or digestive tissue and see its effect. So the presence of a CUGBP binding site, which is a feature, in the upstream intron leads to increased exon inclusion in central nervous system tissue, whereas it leads to increased exon exclusion in digestive tissue. Okay, and there's the color key over there. All right, so again, we won't go into details here, but the overall code, or the overall map, if you like, takes up sort of 4 or 5 pages, just a longer version of this. Oh, there's the transcript structure features I was telling you about before. So things like exon length, it has an impact on tissue-dependent splicing. Yeah?
But how do you account for feature correlation? If you have multiple correlated features, wouldn't your weights be somewhat misleading as far as the feature importance? Because
yeah, that's a very good question. I don't really have time to go into it right now, but you're right. So if you had 2 features that were nearly identical, you might find that they both score highly, whereas really maybe one of them is redundant. Yeah, it's a good question. I won't even answer it.
Can you talk a little bit about how the field values the interpretability versus the accuracy? Like if you could double your accuracy, but you couldn't tell at all what the model was doing, is that a useful thing, or is that not useful at all?
Oh, I think it is useful. So medical researchers are interested simply in whether or not a particular exon is regulated in a specific way, say in neural tissue if they're interested in a neurological disease. And so then they're just interested in prediction. Yeah. Yeah. On the other hand, if you're trying to find out whether a particular SNP, or single nucleotide polymorphism, a mutation in a regulatory element is responsible for the disease, then you want the interpretability part of the code to be accurate. So you want to know that there really is a binding site there and that there's a mechanism, a biological mechanism corresponding to that binding site. Okay, so I'm going to finish up by talking about a few predictions that we validated in the lab. Well, I'll skip the first one and show you the second one. So this is, we actually validated predictions in 2 different ways. We used microarray data and also Rt-Pcr experiments. And the Rt-Pcr experiments are sort of the more trusted ones, so it's actually good that I can show you this particular slide. So what we did is we took 14 different exons that we had not analyzed using that we had not used to train our code, and we made predictions for those 14 different exons in these 4 different tissue groups. And then what we did is for each pair of tissues, we measured the change using Rt-Pcr. So we measured the change in percent inclusion for those 2 different tissues. And that's what we plotted on the x-axis here. So for example, green is central nervous system tissue versus embryo. So a value of minus 50% means that, for example, this particular exon was more highly included in central nervous system tissue than it was in embryo, and the difference was 50%. Then we did the same thing using our code. So we applied our code to those exons and made predictions for what would happen. We came up with these probabilities, p, and then we just computed the difference in the probability, delta p. And that's what this plot is showing you. There's a relationship between the predictions and the measurements. We excluded from this plot all predictions that had low confidence. So if the delta p was near 0, that means the code wasn't really sure whether there would be a change, so we removed those predictions. And so that's why you don't see any data in the middle here. And we get pretty good agreement. You can see the correlation looks good. And if you compute an accuracy just by putting a threshold in the middle, you get 93% accuracy in terms of predicting the direction of change. We also looked at, to answer your question before about the usefulness of the code in terms of trying to understand how regulation occurs, we looked at a specific example, which is DAM1, not DSCAM, actually this is DAM1. It's a gene that's conserved in different species. It has at least 11 isoforms, and it seems to function in cortex tissues. So we took a look at this, gene, and we looked at the upstream intron. So there's a particular exon that's known to be alternatively spliced. So we looked at the upstream intron, and we focused in on this region and asked, what does the code say about how this exon will be regulated using features in this upstream intron? So if you look at just crudely speaking, if you look at the gray bars and the red bars, those tell you what the code thinks is going to happen in terms of the regulation of this exon. So those tell you that if there's a particular feature in this region that the code has associated with changes in that exon. So these are the regulatory elements predicted by the code. So what John did is he created mutants, a set of mutants, 15 in total. Each mutant consists of this version of the gene, but with a subsequence replaced with random sequence. So you take the subsequence here, flagged by 1, and just replace it with random nucleotides so as to get rid of the regulatory element. Okay?
That's not experimentally? Or
Experimentally. Yeah, so the map, if you like, or the feature map was computed computationally, but John carried out wet lab experiments, about 3 months of work, to make all of these mutants and test them in the lab to see how they would influence splicing. Okay, and so here are the results. These are gels. If you don't know what gels are, don't worry about it too much. Here's the percent inclusion values. So here's wild type. This is the normal version of this gene where nothing's been fiddled with. So we get about 66% inclusion. And then if you look at these different mutants, for example, mutant 1, the inclusion level is 83%. So indeed, this sequence here does appear to have a regulatory effect. And if you look across these results, you'll notice there's only one of them that's very close to wild type, which is mutant 7, which has an inclusion level of 65%. And mutant 7 was predicted to have no effect. Okay, so that was our predicted negative.
You only had one?
Yeah. Well, so yeah, we had to play this game here. The biologists were interested in this example because it was predicted to have a lot of regulatory features. So they thought it was interesting. Of course, that means analyzing the negatives is hard. So the p-value is kind of like 1 in 15, if you like, right? Because there's one negative and we got it right. Yeah.
Yeah. So how do you know all the features you have are sufficient? There could be a lot of
Oh yeah, yeah. That's a good question. And in fact, that curve I plotted of the code quality, it sort of achieved a certain level. And one question you could ask is, what's the maximum theoretical level? And it's about 100 times what we achieved. Now the problem is that we don't know how much of that is due to noise, right? So, if you're flipping a coin, it would be nice to be able to predict every toss. But if it's just noise, you can't predict it, right?
How about possible mismatch between training and testing?
No, we were very careful to sort of test it in all sorts of different ways. So we used cross-validation, standard machine learning cross-validation. And then we also, as I said, used a method where we tested separate exons using different experimental assays. So not using cross-validation, but a completely different experimental assay.
Your experiments, when you did the combinations 9, 10, 11, and 9, 11, for example, those are a bit, I guess that would be the interesting part, really.
Yeah,
how
it is.
The combinations, like 1, 2, 5, 8 doesn't seem to do better than 2 alone.
Yeah.
9, 10, 11 does seem to really make a difference, but then 9 and 11, not so much. So
yeah, so right. So if you look at the code, it'll actually tell you, for example, there's CU elements corresponding to kind of PTB binding tracks. And the way the code predicts that those elements will work is that if you have fewer than roughly a dozen, then you don't see much regulatory effect. If you have more than roughly a dozen, then suddenly you see a regulatory effect. So it seems like those elements work synergistically, that they cooperate together to lead to a regulatory effect. And then the code also predicts antagonistic effects. So there are some elements in this region here, in fact, like you pointed out, where one element will have a positive effect, another element will have a negative effect, and when you produce the combined mutant, then you see the combined effect of removing both of those antagonistic features. Yeah.
I'm having trouble understanding the information for like 6 and 3. So are those examples of false negative in the system, meaning that...
6 does have the gray bars here. It's just weak. Yeah.
Okay, so unbiased motifs are the prediction of the system rather than the red.
Yeah, so so we're kind of over time, but I'll very briefly answer that question. So the red blocks are the features that were from this compendium of 1,014 features. The gray blocks correspond to a de novo motif search that we performed. So just throw away all of the literature, throw away everything, just do the obvious computational thing, and just look for enrichment of motifs, and then we use those to learn the code to refine them, and that's what the gray blocks are. So those are sort of just de novo motifs. Yeah. Yeah, as I said, so you could think of the p-value being something like 1 in 15. So, yeah. All right, so I'll finish up just by saying that I have actually produced a software tool. You can go to the website, drag and drop a sequence, and then hit submit, and then we'll make predictions for it. And then you can click a button, and you go to the UCSC genome browser, and you can see how your sequence of interest is regulated, or how we predict it's regulated. And then you can compare that with all sorts of other interesting things like SNPs and other things, other tracks on the UCSC genome browser that you think might be of interest for the particular gene you're looking at. All right, so to summarize the contributions here, I think one of the things that I was most interested in is just formalizing the problem, right? As Lee was saying, so this is, we actually have a training set. Yes, we have a training set. We have a MATLAB matrix. You can download it and try out your own technique. And then the new biology, I think, was really exciting. So we actually made several predictions, several of which I didn't have time to talk about today, that involved mechanisms, biological mechanisms. And we were able to test, our collaborator was able to test those in the lab and verify them. So that was really neat. And also this notion of actually making predictions is novel. So people have not made predictions for how splicing will occur in different tissues before. They've drawn sort of cartoons, like you've probably seen these cartoons where there's sort of blobs, which are proteins, and there's arrows indicating interactions, but they've actually not been able to make predictions. So we actually made predictions and were able to plot curves. We also looked at this DAM1 example, which I showed you, plus 4 other exons, which I didn't talk about, and validated these regulatory programs or regulatory elements. And then finally, we introduced this computational tool for assembling the splicing code, which I think is sort of interesting, but I think now that we've set up the problem, everybody in the room here could come up with a better way of doing it. And also this web tool, which is also novel in the community. So actually having a web tool, you can literally drag and drop a sequence and then click enter and see how your sequence will be spliced. All right, thanks.
I thought the most important, interesting thing is that you actually come up with best set of features that enable you to
yes.
So
right.
Was it many of those were known before? Or you can
about 170 were known, and the other 800 or so were novel. Yeah, but you're right. You're obviously familiar with how these things work, right? The feature. So Josef Barash came out with a feature set, and yeah, that's from my point of view, that's most of the value of the work. Yeah. No, so there's a set of about 60 transcript structure features which people hadn't really characterized before. So we were the first to sort of write down a description of what those features mean and how you could quantify them, measure them. And then aside from that, about 600 of them were these novel motifs, novel subsequences.
Are these features difficult to measure, or are they?
Well, no. Once so Josef has probably about 24 Perl scripts that he uses to extract the features. So it's complicated. Yeah, it's just complicated to extract them because they're they're not homogeneous, right? They're things like exon length. You'd have to compute that differently than you'd compute something like secondary structure or splice site strength or whatever.
They are coming from the raw data.
Yeah, the raw sequence. Yeah, literally you just input the RNA sequence, and then his set of scripts extract all of the features.
So how will just the secondary structure plus conservation?
Yeah, that's a good question. So if you just look at conservation,
seems to be doing fairly well.
Burge's lab and Ask's lab have shown recently that conservation seems to be indicative of neural specific splicing. So if you just look at where did my, oh, it's over here. Yeah, if you just look at conservation only, it's a very good question, yeah. You do nearly as well. But you don't do as well. And in fact, if you take, what's also interesting is if you take our final inferred code and keep all the features except those that use conservation, we do about the same as if you use only the conservation only. So I think this gap here is probably quite significant in terms of what's going on.
But what
It's
comes
bio-
from the graph is conservation plus structure, which will still not be very specific of the alternative splicing or any learning, but it will just be structure.
Yeah. Yeah, I don't know.
No motifs though.
Yeah, we haven't done that.
Because I'm wondering how much of your motifs will just indicate in structure.
I see. I see. Yeah. It's possible that, yeah. Yeah, we should try that. Yeah.
I'm still a little curious about the way you set up the problem. So you set it up so that you're predicting whether it increases or decreases, not by the amount. Did you start by trying to predict the amount and then it was too noisy?
Yeah, we tried to predict the amount for about a year and couldn't get anywhere.
And why was that? The amount, so you think that the amount itself is just really hard to predict, it's just whether
Yeah.
up or down.
So the best way to think about why that would be the case is there are just a lot of unknown variables that we didn't measure. So, for example, there might be, and actually if you look at the splicing code, this is something people always ask about. If you look at FOX in central nervous system tissue in the upstream intron, it seems to lead to both an increased inclusion and increased exclusion. How can that be? And the reason is that there might be another protein that interacts with FOX. So if that other protein is present, you get increased inclusion. If that other protein is absent, you get increased exclusion. And so you can actually basically account for a change either way using this additional hidden variable. So that's the issue, is these other hidden variables that we haven't measured.
But in this case, when you measured, let's say those amounts up and down were equal, you would have measured the no change, and you'd be trying to predict no change.
Yeah.
Is that right?
So basically we see that CNS is going to go one way or the other. We can't predict which way it'll go, but it's definitely not going to stay stay at the status quo. It'll go one way or the other. Yeah.
And the data that you have available actually has the amounts, or does it just have these sort of three?
It has the 4 probabilities, well, 12 probabilities, 3 for each of these tissue groups. Yeah, I mean, you could take, if you want, so the original data was published in 2007, and it has the % inclusion values for the 27 different tissues. So you could take a look at that if you want to try predicting the actual level. I think, ultimately, I think that's what one would like to do, right? It's just that the model will have to be a little bit more complicated. You'll have to have some additional hidden variables to account for the absolute level of the change.
I don't value that it's very hard and almost impossible to do. I'm just curious if that data is available for, like, you know, a biologist or whoever are going to try to.
Yeah, no, I don't think it's impossible. I think now that we've done this first step, you could just imagine a system that contains multiple components, right? The first step is to predict whether you're going to get a change this way or that way. The second step would be to predict how great the change will be. So what we did is just focused on that first part. Yeah. Yeah.
You initially mentioned that the final method that you used is somehow got a physical correspondence or implementability by the brain.
And when you say implementable by the brain, you mean interpretable?
Yeah,
Sorry.
yeah.
Or you say, okay. Yeah. So when we show this to the biologists, they love it. This code is very interpretable to them, and it actually, in many, so first of all, the biologists are familiar with maybe 20% of these predictions, and they correspond to what they know. So the table makes sense to them. The table probably contradicts maybe 10% of what they know. So those are interesting examples that people are following up on to find out whether it's a mistake in our table or a mistake in the current understanding of what's going on. And then for the other 80% of the predictions that they don't know about, various people are now trying to understand those in more detail. So they're trying to study the mechanisms that are predicted by the code and see if they can actually measure, for example, proteins. So the code says that there's a particular protein in a tissue that'll have an effect, and nobody's observed that before. So now the biologists can go into, they can set up an experiment where they actually measure the abundance of that protein, and then they knock down the protein and see if they knock down the protein, does it have the predicted effect in that tissue.
So that kind of will actually elucidate what kind of hidden variables.
That's right.
That might eventually
Yeah, it's sort of
better prediction.
Exactly, a feedback loop. So we'd take whatever result they get in terms of these additional hidden variables, throw them into the code, and improve the code quality. Yeah.
Are your tissue groupings pretty standard or accepted in the biospace?
No, that's actually a good question as well. So we have these 4 tissue groups. We extracted them automatically. I didn't have time to talk about how we did that, but it's sort of a machine learning, an unsupervised machine learning technique. And we found one group that people hadn't looked at before, which is this digestive tissue group. And we're able to make predictions for it. So it's not just sort of a noise group. We can make predictions and then validate the predictions. So there's a novel group there. And also we discovered two subclasses of cortex tissues, which I didn't talk about today. But we're able to sort of predict differences between these 2 subclasses. Ultimately, I'd like to be able to, of course, make a prediction for all 200 human cell types. And
I wonder if some of that grouping might influence some of the ins
Yeah.
and outs that you're
Yeah, I think it does. Yeah, I think it does. Yeah, in fact, when you see a feature having an effect in both directions, it could be that that's a grouping of 4 or 7 central nervous system tissues, and there's a subset of frontal lobe tissues where there's an effect in one direction, and then there's a subset of hindbrain tissues where there's an effect in the other direction. Yeah. The problem is that the data's more like this, so even if we found something, it might be spurious. Yeah.
It's just curious.
Okay.
In terms of talking about feature extraction, so the raw data is coming from genome sequence to begin with.
The feature part, yes, that's right.
So now, when we do speech, now there are a lot of people actually thinking about from the trial group, they treat the waveform as a raw data.
Right,
and
right,
you
yeah.
can do all layer by layer learning out there.
Right, yeah.
Learning stuff.
Yeah, yeah.
So do people think about this way of doing,
So my student, so I have
a
a young new master's student who just finished his coursework, and he looks at our feature compendium and says, "Oh, that's too complicated. Just give me the raw sequence." And so he tried that, and and I was really impressed because, as you say, he's only 8 months into the project. So he tried that, and just looking at the raw sequence data, just looking at the raw sequence data, he gets a result that's somewhere just a little bit worse than the known motifs. So, right, yeah. I mean,
So that has done it. This is just the raw data.
He just literally looked at the raw genomic sequence and then used kind of a neural network technique. It's naive, but it's impressive that he was able to do nearly as well as what you'd achieve if you used all the prior literature in the area. Right. So what he's going to try doing now is the kinds of things he did with speech, like a convolutional type of structure. Instead of just taking all of those nucleotides and feeding them into a neural net, he'll use kind of convolutional neural net. So you sort of sweep it across. And
people actually use the layer by layer structure to do that.
Yeah, you can do that too.
More reason.
Yeah.
For that kind of thing.
All right, shall we finish it? Thanks.